o'clock. So thank you very much for joining. I'm Michael Holland, Associate Director of Corporate Finance at Sequoia. We have been mandated along with our friends at Tech Voyage to help Graham Kelly, CEO of Filamon, with their pre-IPO capital raising. So we are raising $3 million to pursue a number of initiatives that the company will undertake prior to an expected ASX listing in the first half of 2025. Over the next 20 to 25 minutes, Graham will run through a presentation, after which point there'll certainly be time for Q&A. If you have any questions as we go, please feel free to put them in the chat and we will put them to Graham at the end. Without further ado, I'll hand it over to Graham, who obviously has experience of 4 ASX listings over his career and more than $350 million worth of capital raised. So he's a hugely experienced ASX professional to take it from here. Thanks, Graham.
Thanks, Michael. Well, thanks everyone for joining in. Look, Filamon is an interesting story. In fact, it's quite fascinating, which is why I've got myself involved in it, because it's tackling one of the biggest problems facing medicine, which is the need for a whole new generation of anti-inflammatory drugs to face what's a coming tidal wave of chronic inflammatory diseases as the baby boomers all hit their 60s and 70s and so forth. Now we're not talking about anti-inflammatory drugs like Panadol or ibuprofen or Nurofen. They're far too simple. What we're talking about is far more potent, more powerful prescription drugs that we're going to need to treat these chronic inflammatory diseases, all of which you can see highlighted in yellow here. So this covers the main reasons that 3 out of 5 Australians are disabled or will die from these age-related chronic inflammatory diseases. And to be clear, we're not talking about causes of death as part of natural aging. What we're talking about are diseases that prematurely kill people because they're effectively causing accelerated aging. Now, the biology of all these diseases is very complex. They're diseases that go on over years, that are response to the wear and tear that all of us get during our life, and they typically involve dozens, even hundreds of genes that are all misbehaving in a way that causes all these inflammatory processes. Now, the problem with the current batch of anti-inflammatory drugs is they're far too narrow, far too limited in their targeting. If you're using them for something like acute asthma, they're terrific because acute asthma is an acute inflammatory disease that's got fairly simple a number of genes involved. Once you get into these chronic inflammatory diseases like heart disease and cancer and dementia, you're talking about symptoms and processes that are just far too complicated for these current drugs. Mind you, the current batch of anti-inflammatory drugs of the type that we're talking about currently sell around about $100 billion a year. So despite the fact that they're only working in very, very few patients who fall within this yellow group up here, they're still doing $100 billion a year. So it's you don't need me to tell you the sort of numbers that would be involved if you can come up with some drugs that will actually do more than what's currently being done now. Filamon is focusing on this new generation of anti-inflammatory drugs through their ability to target multiple genes and multiple inflammatory processes. And these are processes that are common to all the diseases that you see up here in this yellow group. So our aim is just a new generation of anti-inflammatory drugs that are more potent and will produce more longer lasting effects than current drugs for the common age-related diseases. So what distinguishes us from what appears to be anybody else that we're aware of, but not just in Australia but worldwide, is that our drugs target these multiple inflammatory genes. They can switch off dozens of genes with the one drug, which is really a key point of distinction. They also need to be well tolerated. The current anti-inflammatory drugs do have side effects, but it's important for drugs to be used on a say 20 or 30 year basis to see someone through to the end of their life. They need to be very well tolerated, which ours appear to be. They also need to cross the blood-brain barrier, because many of the diseases we're talking about are diseases of the brain, diseases like dementia. And they also need to be oral, whereas the current anti-inflammatory drugs are almost all given by injection. So these excuse me, these are the features that we've sought with our drugs. I'm not going to go into too much detail, but we have 3 platforms, and they each target a different inflammatory process. And these processes are common to all these diseases. And so we're simply producing drugs that we will mix and match for particular disease states. Now, instead of regarding this problem as tissue-centric, that is it a disease of the brain, or is it a pancreatic cancer, or is it a breast cancer, or is it rheumatoid arthritis? We see the disease as the chronic inflammation. That's the process that underlies all these diseases. Which means that if you treat it that way, if you look at the world that way, then your drugs that you produce have the potential to be used across multiple forms of chronic inflammatory disease. But we're just simply that's a massive choice we could take on. But we've decided to focus just on 3 diseases, which are the ones that really are arguably the most pressing, facing this baby boomer generation. And that's dementia of the brain, wet macular degeneration, which is the major cause of blindness in elderly Australians, and all forms of solid cancer. Now, today I'm just going to focus on the eye and the cancer, and we'll just leave the brain because it's a complex story. So starting with cancer. Now, cancer remains one of the top 3 causes of death of elderly, and causes an estimated 10 million deaths worldwide each year. Now, it remains a lethal disease despite all the undoubted improvements that have come over the last 20 or 30 years. It remains a lethal disease because despite all those improvements, once the cancer becomes aggressive and spreads, patients end up with very few effective treatment options. And despite all these new whiz-bang developments in chemotherapy and radiotherapy and immunotherapy, where they do work, essentially what they're doing is delaying the inevitable, not preventing the inevitable. So the question is, with all these big improvements, why haven't we made this quantum leap into being able to do something more than just delaying the inevitable? And the evidence is increasingly pointing to something called the tumour microenvironment, or TME, and that this is maybe the missing piece of the puzzle. And current anti-cancer drugs have little or no effect on this tumour microenvironment, which is protecting the cells from treatment. And it serves to nurture any cancer cells that might survive the treatment. And it doesn't matter how good a treatment is, it's extremely difficult to kill all the cancer cells. Some will always survive, and it's those surviving cells that the tumour microenvironment nurtures and brings back, often in a far more aggressive form. Now, our drug KSC2, in our view, looks to be the first comprehensive inhibitor of this tumour microenvironment. So just to explain what this is. So here you have a mass of tumour cells. And in that tumour, only roughly half of all the cells in any tumour are cancer cells. The other half are made up of all the other cells that you see around any sort of tissue. Lots of blood vessels, something called mesenchymal cells, stem cells, which are incredibly important for how a cancer spreads. Fibroblasts, these fibroblasts produce collagen, which binds everything together, as well as providing an actual barrier that makes it very difficult for chemotherapy drugs to infiltrate into a cancer. Immune cells and even fat cells. So on the surface, these all look normal cells, but in fact, they've all been hijacked by the tumour cells, and to create what is this tumour microenvironment. And what happens is that this is created by something called epithelial mesenchymal transition, which hardly anyone outside of science has ever heard of, but this is a critically important process. It's how your body heals when you get a cut or damaged tissue that needs to be fixed up in a matter of a week or so. This is the process the body uses. It's the process it uses as a fetus, as an embryo, to be able to corral all the cells to grow quickly, to form an organ. So it's a very, very fundamental process. It goes back to if a squid or an octopus loses a leg and wants to grow another one, this is what it uses. And this is the process that cancer hijacks to create this tumor microenvironment. So this is the end result. This is the process, and vimentin is the protein that drives it all. So there's been a rush to find a drug that will block vimentin. The problem is vimentin is so crucial to all our cells in our body that once you start knocking it off in a willy-nilly fashion, you're inevitably going to end up with an enormous number of side effects. So we believe the KSC2 is the first inhibitor of this protein when it's inside a tumour. So in other words, we are able to contain the side effects to those of the tumour. And that is what got me interested in this company when this technology was first shown to me by my two co-founders. I realised that they had really identified something that nobody else has been able to do. So that's why I'm involved. So KSC2 has already undergone a first-in-human study. In fact, when my co-founders, such as Professor Paul D'Souza, came to me some years ago, they'd already done a trial. They'd already taken this drug into humans. And because it was the first vimentin inhibitor to be used in humans, there were some very large questions over how safe it was going to be. So they had to conduct that trial very carefully and with some degree of caution, but they conducted it in 12 patients who had advanced prostate cancer, and all those patients tolerated the drug very well. So we've pretty much answered the very first question you have to answer with any drug is it safe? Now, the second question you have to answer is, does it work? And that's what we're about to do. And this pre-IPO capital raise we're conducting is designed to give us the funds to at least get this study underway. And it's a phase 1, phase 2 study. It'll be conducted in up to 80 cancer patients. These patients will all have late-stage cancers that's progressing on standard of care, whether it's chemotherapy or immunotherapy. It'll be what's called an all-comers trial, which means that we take anyone who's got any form of cancer, providing the cancer is progressing on standard of care. And we see this advice study as being a world first study, the first time anyone's been able to look at using a drug that blocks the actual causes of this tumor microenvironment in conjunction with standard anti-cancer therapy. So we believe that this trial will be watched widely and with great interest around the world. Now, the second major drug that we're testing is a drug for this eye problem called neovascular age-related macular degeneration. As I said earlier, this is the major cause of blindness in older Australians. Here's the retina, the back of the eye. This macular region on the retina is responsible for central vision. And macular degeneration, you can see here, is just this profusion of blood vessels that grows, and you get lots of fibrous tissue. So it's classic chronic inflammation. And light cannot penetrate through it to the back to the retina. And what patients lose is central vision. So they see just a black void in front of them with a little bit of peripheral vision around the edges of it. But that's a progressive disease that in most people leads ultimately to total blindness. And the treatment currently that's available is an injection. It has to be injected into the eyeball. The problem is that, like all current anti-inflammatory drugs, no matter whether they're used for this condition or rheumatoid arthritis or anything, they're very limited in their action, and in fact they don't work in 1 in 3 people at all. Where they do work, they don't stop the disease; they simply slow it down. It certainly is an uncomfortable treatment where you have to have the drug injected directly into your eyeball, and it has to be done monthly for the rest of your life. So the federal government and the University of New South Wales are helping us develop this drug, LKBT2, as a breakthrough treatment for this disease, where it's going to be used as an eyedropper. So between the government and the University of New South Wales, they provided us with almost $5 million to get to this drug to the clinic. So effectively, this is any investor in Filamon at the moment is getting a free kick. This is money that has been given to us to bring this drug into the clinic. And the reason that people are excited about it is that it knocks out all these different genes. These are genes that the other current drugs cannot hit. So we're getting this broad effect, which in animals has already shown that it's up to 3 times more effective than the current lead drug, aflibercept, which currently has sales of close to $15 billion a year. And but as well as being a broader in action than the current leading drugs, it's also small molecule. These current drugs have to be injected into the eyeball because they're large molecules, they're big, they're antibodies. What we do, our drug is being developed at the moment as an eyedropper. So, it's quite obvious that if this works as well as, or even better than the current drugs, plus has the ability to be self-administered, you don't have to go and see the ophthalmologist every month, and if it works in more patients, then it stands a pretty good chance of becoming the market leader. So this is a unique opportunity that the federal government recognised, and given the significant cost in caring for fully blind people, that is going to, anything like this that would stop that from happening would be have a major impact on one of the major costs of care of elderly people. So this is a growing problem because, as I mentioned earlier, all us baby boomers are about to hit our 60s and 70s. So hence the term the silver tsunami that's on its way. And this is a problem that's going to go on for the next 20, at least 20 years, until the baby boomer generation is all in the ground. And all these conditions we're talking about are chronic inflammation. All these aging diseases, they all have underneath them the same processes that our 3 drugs have been designed to hit. So we believe that we're well placed to make a significant difference to this this major problem that's coming. So we've got multiple milestones coming, but I'm just going to focus on just the two drugs I've spoken about and when they're going to be in the clinic. So the anti-cancer drug in the advice study, we anticipate will be the patients will be starting to be enrolled hopefully even before the middle of the year, but certainly by the middle of next year at the latest. And that study will then go on for at least 2 years. The eye disease drug, we expect to be in the clinic towards the end of next year or beginning of 2026. And at the moment, this drug is having its development paid by the federal government via a large French company that specialises in drugs for the eye. Our IPO is targeted for this area here in Q1 or Q2 next year. Our ideal target would be somewhere here round about March next year. But clearly, that's something that's going to be driven by market conditions and the ASX and when they're ready for us. So just briefly about the 3 founders. The drug for the primary drug for cancer came out of the brain of this scientist, eminent scientist, Kieran Scott. He started the development of it when he headed the inflammatory diseases unit at the Garvan Institute of Medical Research, which is part of the University of New South Wales. He's now a professor of oncology in Western Sydney University. He associated himself with Paul D'Souza, who was one of Australia's most eminent medical oncologists. Paul currently holds the chair of medicine in oncology at the University of Sydney. He has spent some time in industry, and Paul has run and been part of numerous clinical studies. For myself, I've started and founded and floated 4 companies. 2 of them are still listed on NASDAQ and 2 on the ASX. So my background really is a mixture of science and entrepreneurship. We have one of the guy that does all our drug design, Jun Zheng, a Melbourne-based scientist. Jun's main claim to fame was that he was one of the designers, perhaps the main designer of this drug, which is now the most successful Australian drug in terms of being bought by industry. So it was sold recently for $1.9 billion to GlaxoSmithKline. So he can put his hand up as the major designer of that drug. So he is now designing all our drugs. Philip Marshall, another one of our directors. Philip has spent most of his life with Mayne Pharma, a very hands-on role in manufacturing and in intellectual property. So Philip is very much hands-on in Filamon right now. So that's the team. We believe that we have all the necessary abilities to make this happen. I certainly have started in enough companies to know how to get a company moving and off the ground. So we have 5 programs underway. I've only told you 2, but there's 5 programs underway. there's going to be a steady stream of news flow starting right now and through to the time of our IPO and beyond. These are all massive markets. We're not talking about markets where there might be 5,000 people a year worldwide suffering the condition. We are talking about 3 of the 5 major causes of disability and death in the world. We've been incentivised to make this work and to show our confidence. What we've done is taken 60%. These are the 3 founders. We've taken 60% of our founder shares off the table and converted them into performance shares. And these performance shares have 2 milestones. They need to have a technical, scientific forward movement. So in other words, they have to go into a clinical trial, which is evidence that to get a drug into a clinical trial, you've got to convince doctors and hospitals that this thing is more likely to work. So that's the first milestone that has to happen. The second milestone is that the value of the share price on market has to be at least 50%, up to 100% premium over the IPO share price. And so we'll have at a $3 million raise, we would have a post-money valuation of $9 million, which, as pointed out here, that represents less than 1/5 of the money that's been spent on this technology to date.
And just to chime in there, Graham, okay, perfect timing with the capital structure. So as you can see here, there's currently 66 million shares on issue. Now, once the founders convert 60% of their holding, that drops to 26 million. Then we're raising 3 million at 22 cents, which creates another 13.6 million shares on issue for a pre-money of 5.7 and a post-money of 8.7. So with that pre-money of $5.7 million, I mean, effectively, that's basically the value of the UNSW and federal government funding of 4.9 million that's going into the company right now for the ophthalmology works. So I think it's an incredibly compelling valuation. The primary purpose of this round is to manufacture a clinical trial batch of KSC2. So Graham touched on it briefly, but one of the key factors that the ASX are looking for to facilitate a listing onto the exchange these days is an imminent commencement of a phase 2 trial. So they need the funds currently to manufacture the dose, which will be sufficient to conduct a trial of between 60 and 80 patients across the country. We've already done the phase 1 for the KSC2 across 12 patients, and this will be a second phase 2, should I say. Pleased to say that 50% and more than 50% of the funds have already been committed, and so we're in a position where we're hoping to close the raise by the end of next week. We can obviously see there the use of funds for IPO as well. That is all subject to market conditions at the time, obviously, but that's a rough idea of what we're thinking it will look like down the track. Graham, you've covered it tremendously. Are there any questions at this point in time? If you would like to ask it, I can open microphones. Otherwise, if it's easier to put it in the chat box, please feel free to do that. Yeah, Jeff, I will just put your mic on. One sec. All right, you should be able to unmute yourself now, mate. Sam, same with you. You should be able to unmute now.
Yeah, hi Graham. Good presentation. Just 2 questions. One's regarding the blood-brain barrier. So my understanding is that is a big issue with the current drugs out there targeting dementia, as far as efficacy is concerned, that they just don't get across the blood-brain barrier. In the presentation, it mentions Filamon can cross the blood-brain barrier. What is that based on? Like, what sort of data do you have on that?
Right. Well, that's based on animal data.
Yep.
So we've treated mice and then sacrificed them and look at the brain. Very standard stuff, and the drug certainly gets into the brain. We believe at levels that would be meaningful to deliver a therapeutic effect. You're quite right about. I mean, current anti-inflammatory drugs, almost overwhelmingly, not totally, but mostly, big antibodies. They're big proteins, and they just can't get across the blood-brain barrier. What our drug is a small peptide. It's got the same molecular size as what's called a small molecule drug. So it does cross.
Is there a percentage that crosses, like that from that mice study, is like 10% gets through, or do you have that sort of figure, or?
Yeah. Well, all you can do is to compare it to other organs, and if you look at most of the drug, when you give it to a mouse or to a human, ends up that most of it goes to the liver, then to the kidneys, and then out in the urine. But if you looked at I don't know, let's say say a bit of muscle, what's in the brain is equivalent to that. So it's not charging across like a hot knife through butter, but it is present in the blood. In the brain, at a level that we know in vitro works on cells. So that's why we believe it would be effective in the brain.
Okay. And just one more quick one, with the phase 1 that you did, that would have been safety related, I'd imagine. Has there been any efficacy results on the animals that you've tested?
Well, on the animals, yeah. I mean, we were able to do the study because when it was used in animals, it had a dramatic effect. All the work in animals was done on prostate cancer, because prostate cancer is one of those cancers that we know inflammation plays an important role. It does in bowel, breast, and particularly pancreatic cancer as well. But all the lead up work was done on prostate, so that's why they did prostate cancer patients. Look, phase 1 is never meant to. The only question you really ask in phase 1 is it safe? Is it well tolerated? If you see some signals that it's working, well, that's bonus. This trial was only done over 6 weeks because the cost of making the drug was so great. Paul and Kieran did this off their own bat. I mean, it's really an interesting story in drug development. They begged, borrowed, and stole to get this trial done, and good on Liverpool Hospital in Sydney, which is one of the biggest cancer clinics in Australia now. They were prepared to run it, and but they can only do it for 6 weeks. So that you're not going to see any benefit after 6 weeks.
No.
However, what they did see is some changes in three out of the 12 men, even after 6 weeks of treatment, showed falls in their PSA level. Now, we're not going to overinterpret that. All that means is that we're encouraged by it, that we would see anything like that after 6 weeks.
Sure. Thanks, Graham.
Thanks, Sam. A couple in the chat, Graham. So firstly, would KSC2 be used as a preventative measure to prevent the inflammation that causes the disease, or more as an initial treatment once disease has been diagnosed?
Yeah, look, preventative drugs being allowed on a preventative basis is a real hard. That's an Everest mountain to climb. I'm not aware of any that have been approved for that. You've really got to be diagnosed with a condition and then treated with that drug. But the earlier you start treatment, then the better. But also, by the whole idea of the drugs we're working on is because they work on multiple genes, they should be more able to stop the disease. Whether they're going to cure it, we'll wait and see. But certainly by stopping the disease and not letting it get any worse, by starting treatment as soon as you're diagnosed or early in the disease, that's the sort of next best thing to having a preventative treatment.
Beautiful. Secondly, I can answer this one. IPO plans in terms of number and shares of valuation. Everything's subject to change, as we always know, right? And obviously the milestones that the company can prove and progress up until that point. We would initially anticipate a share price circa 35 cents and a $12 million raising. So that puts you at about 73 million shares on issue post IPO with a market cap of circa 25 or 26 million is roughly where we're thinking currently. Just beautiful. Thank you. Okay. Just conscious of everyone's time, unless there are any final questions. Oh, no, there's one more. Sorry. Presumably treatment would be together with primary standard of care as the trial set up.
Yeah, exactly. Yeah, so it's not intended to be a standalone drug. It's a drug meant to be used in any form of cancer therapy.
Beautiful. Thank you very much, Graham, and thank you very much, everyone, for joining. We'll obviously get back in touch with you over the next few days. Should you have any questions following up the call, we're more than happy to answer either Graham or myself, and we can distribute this as a video should you wish to watch again or distribute yourselves. Thank you very much, everyone, and have a lovely day.
Thank you.